Welcome back to the deep dive. Today we are opening the door to perhaps the most ambitious feat of bioengineering in the lab right now. Recreating the entire human digestive tract cell by cell in miniature.
Right.
We are talking about the small intestine, a masterpiece of complexity.
Yeah.
I mean, it's not just a tube. It's this sophisticated, multifunctional chemical factory. It's responsible for nutrient absorption, maintaining a selective barrier against, trillions of microbes, dictating metabolism. It's just incredible.
It is truly mind-bogglingly sophisticated. And yet, for decades, if you were a drug developer or, a researcher studying a specific intestinal disease, you faced a significant hurdle. You either relied on incredibly invasive, difficult-to-scale human biopsies, which are, of course, finite, or you had to rely on these simple laboratory models that were, well, fundamentally broken.
Yes, exactly. Our mission today is to dive deep into a microgut revolution, one driven entirely by induced pluripotent stem cells, or iPSCs. We have been poring over two really significant pieces of research detailing how scientists are leveraging the power of these iPSCs to generate radically superior gut models.
And these models, they come in two complementary flavors, which is what's so cool about this.
Right. You've got the super pure 3D human intestinal organoids, which we'll just call HIOs, and then you have the highly functional, scalable 2D epithelial monolayers, the HIECs.
This is really the moment where basic science moves from these crude approximations to highly patient-specific functional replicas. I mean, these models are redefining how we study everything from personalized medicine.
Like finding the right treatment for a specific individual with cystic fibrosis?
Exactly that. All the way to predicting whether an oral drug will actually work in the human body. It's a huge leap.
Okay, so let's unpack this by first grounding ourselves in the past.
Yeah.
If the small intestine is this high-tech biological factory, what was the standard, the outdated tool researchers were forced to use until this whole iPSC revolution began?
The decades-long standard, really the workhorse of gastrointestinal and drug absorption research, has been the CACO2 cell line.
CACO2.
Yeah. And to put this into context for you listening, CACO2 cells are human colon adenocarcinoma cells. They're a cancer line.
Okay, cancer line. And from the colon, not even the small intestine.
Not the small intestine. But they've been used so extensively for one really simple reason. They spontaneously form a relatively organized layer when you culture them on a flat plate. So that made them technically useful for these initial, very basic studies measuring how molecules move across a barrier.
But right there, that basic mismatch, it should be setting off alarm bells, right? We're trying to study the small intestine, the primary site of absorption and metabolism, and we're using a cell line from a cancerous distal organ that mostly just reabsorbs water.
That sounds like a severe limitation before we even get into the details.
Oh, it's more than severe. It creates two huge functional deficiencies that they severely compromise the relevance of the whole CACO2 model.
Okay, let's start with the first one then. Cell diversity.
Right. So the human small intestine in vivo, in the body, it's this dynamic ecosystem built on the collaboration of four major epithelial subtypes. You have the absorptive enterocytes. These are the cells CACO2 sort of tries to mimic, the ones responsible for pulling nutrients and drugs across the barrier.
Okay, so CACO2 has a version of that one.
A very simplified version. But then you have three absolutely crucial secretory cell types that CACO2 either completely lacks or produces really poorly.
And which are those critical missing elements? First, the goblet cells. They produce and secrete the protective mucus layer. That mucus is the first line of defense, and it completely dictates how a drug interacts with the surface. Second, you have the Paneth cells. They live at the base of the intestinal crypts and secrete these potent antimicrobial peptides. So they are essential for local host defense, for regulating the gut flora.
And
third, the enteroendocrine cells, which are basically the hormone factories of the gut. They're secreting these crucial signaling molecules that talk to the brain and regulate digestion.
So if CACO2 only really offers a simplified version of that one absorptive cell type, you instantly lose the ability to study all these complex interactions, like mucus barrier formation, how the gut defends itself from bacteria, or how specific hormones get secreted in response to a meal or a drug. That's a fundamentally incomplete model.
It is, and that incomplete picture is exactly what makes the model fail on the second crucial point, which is metabolism.
Metabolism.
This is absolutely critical for the pharmaceutical industry. The small intestine isn't just an absorption tube, it's a detoxifier. When you take an oral drug, the very first place it encounters metabolic enzymes is right there in the gut lining before it even gets to the liver. This is what we call first-pass metabolism.
Ah, the famous first-pass metabolism gatekeeper. For those of us who track drug development, we know the enzymes involved can completely dictate a drug's effectiveness.
Exactly. And the most important, the most abundant enzyme in this whole process in the small intestine is cytochrome P450-3A4, or CYP3A4.
CYP3A4.
Right. This one enzyme dictates the fate of a huge proportion of all orally administered medications.
And CACO2 cells. I'm guessing they don't have it.
CACO2 cells exhibit a massive functional deficiency in CYP3A4 expression. It's negligibly expressed, it's effectively absent, and since drugs are so often broken down right there in the gut before they even get absorbed systemically, CACO2 is rendered fundamentally unsuitable for accurately investigating intestinal first-pass metabolism, and so you can't predict real-world bioavailability in humans.
So if a pharmaceutical company uses CACO2 to screen a new drug, they're essentially getting a false positive.
Yeah.
They might think the drug is absorbed perfectly, but in a real human, the gut's CYP3A4 would just chew it up before it ever reached the bloodstream.
Precisely. They are modeling transport without modeling the chemical processing facility that comes with it. This is why drug failures late in development due to poor bioavailability are so common and so incredibly expensive. We needed a model that could provide the cell diversity and the metabolic machinery.
And this is where the power of induced pluripotent stem cells, iPSCs, completely flips the script.
The discovery of iPSCs, where you can take specialized mature cells, like a skin cell, and chemically coax them back into an embryonic pluripotent state, that was the inflection point.
Right.
Suddenly, we had an inexhaustible patient-specific source. You can take a patient's blood sample, turn those cells into iPSCs, and then direct those iPSCs to become virtually any tissue you want, including the intestine.
And critically, the tissues derived this way, they tend to form these complex multicellular structures that look and act a lot more like their in vivo counterparts than any cancer cell line ever could.
Exactly. Now, we did have initial success generating 3D human intestinal organoids, these HIOs, but the source material notes that those original methods had a huge scalability problem. They relied on primary tissue.
Right.
Yes, culturing from primary intestinal crypts, the natural stem cell niche in the gut, that was a breakthrough for sure. But getting those crypts required an invasive biopsy procedure. Every single new experiment or drug screen needed a fresh, invasive sample.
So it just wasn't scalable.
Not at all. iPSCs sidestep this entirely. They offer the ability to produce these sophisticated, personalized tissues indefinitely, which opens the door for creating vast biobanks and supporting truly large-scale modeling.
So the raw material problem is solved. Now let's get into the first major technical breakthrough that our sources present. How researchers use this raw material to achieve a truly pure, highly defined 3D HIO.
Okay, so when we talk about purity in the context of these gut organoids, the critical contaminant that researchers have always struggled with is the mesenchyme.
The mesenchyme.
Yeah, this refers to the underlying connective tissue, the mesenchymal stromal cells, which are supportive elements in the body.
And why does the mesenchyme even show up in these lab cultures in the first place?
Well, because in the natural development of the gut, which is the process they're trying to mimic in the dish, the epithelial layer and the underlying mesenchyme are constantly interacting. They provide essential signaling cues to each other. So the early traditional protocols, often called mesenchyme-containing or MC protocols, they resulted in organoids that contain both epithelial cells and these mesenchymal stromal cells.
And if your goal is to study an epithelial disease, a defect that's confined purely to the lining of the gut, the presence of those healthy mesenchymal cells creates a massive amount of experimental noise, right?
Exactly. Think of it like trying to tune a specific instrument in a full orchestra. If you're trying to study a defect in the epithelial cells, the mesenchymal cells are still chattering away, maybe even compensating for the defect or generating their own confounding data points.
So you had to get them out somehow.
To isolate the epithelial component, researchers had to use expensive, really time-consuming techniques like fluorescence-activated cell sorting or FACS to physically separate the epithelial cells, the EpCAM-positive cells, from the rest of the culture. This was a major workflow bottleneck.
So the mission for the research we're looking at was clear. Develop a mesenchyme-free, or MF, protocol that eliminates the need for that external support or isolation entirely, creating a purely epithelial-only model. So how did they achieve that kind of precise control over differentiation?
This is where the sheer elegance of directed differentiation using chemical cues comes into play. It relies on this multi-step process using specific, defined, serum-free media conditions. They're essentially telling the iPSCs exactly which developmental road to take and, just as importantly, which roads to block.
Let's look at that road blocking step first. How do you stop them from becoming something else?
So the process starts with differentiating the iPSCs into definitive endoderm, which is the precursor layer for all internal organs. Then comes the crucial gatekeeper step, dual SMAD inhibition. They use these compounds, dorsomorphin and SB431542. SMAD signaling is responsible for driving cells down pathways that lead to other organs, like the lungs or the liver.
So by using this dual SMAD inhibition, they're effectively blocking the signals that would turn the cells into unwanted tissues.
Yeah.
It's not about promoting the gut, at least not at first. It's about preventing the formation of everything else.
Precisely. They're closing the door on all the competing fates. This leaves the cells receptive to the next set of signals, the ones that specifically guide them toward the hindgut, which is the precursor to the lower GI tract.
And what chemicals provided that specific direction, that push towards the gut?
They promoted lineage specification using Wnt activation via a compound called CHIR99RO21, or CHIR, which is essential for intestinal identity, along with BMP4, that's bone morphogenetic protein 4, and retinoic acid, or ORDA. This combination precisely guides the endoderm fate toward the intended destination, the hindgut progenitor.
Now, to verify that this elegant cocktail was actually working, the researchers used a brilliant technical trick. They created an internal tracking device.
Yes, the CDX2 GFP reporter iPSC line, which they dubbed BU1CG. So CDX2 is the critical transcription factor. It's the master switch for hindgut and intestinal progenitors. The researchers engineered the cells so that whenever the CDX2 gene was active, the cell would also produce green fluorescent protein, or GFP.
So they could just watch them turn green.
Exactly. It allowed them to visually track, measure, and separate the desired cell population in real time. It's like putting a little fluorescent tag on the specific cell type they wanted.
So what did the tracking reveal? How did the timeline look?
It provided really clear quantitative evidence of success. The CDX2 GFP positive cells, the green ones, started emerging quite early by day 8. And by day 13, these intestinal progenitors represented a significant portion, about 41% of the total cell population.
But the real operational proof of concept came from the sorting. If that GFP-positive population truly contained all the intestinal potential, then the cells that didn't glow green, they should fail to make an organoid, right?
That was the defining test. They physically sorted the day 15 culture, separating the CDX2 GFP positive cells from the negative cells. When they plated the GFP positive cells in the 3D culture medium, boom, they formed robust, healthy, functional organoids.
And the negative cells.
The GFP negative cells failed almost completely. This confirms the vast majority of the potential to form a gut resides specifically in that CDX2 positive population.
So after all this precise chemical steering and purification, what was the quantifiable result? I mean, how pure was the final MFHIO product?
The results were genuinely transformative for the field. The resulting MFHIOs showed virtually no staining for the mesenchymal marker vimentin, which confirmed they had successfully eliminated the stromal cells.
Okay, so visually it worked. What about the numbers?
Flow cytometry provided the hard number. The MFHIOs were incredibly homogeneous, measuring 99.25% epithelial cells, or EpCAM positive.
Wow. Nearly 100% pure epithelial tissue, derived entirely in vitro from stem cells without any need for complex sorting steps. That is a game changer, especially when, as you said, the older mesenchyme-containing protocols might only yield around 50% epithelial cells.
The significance here is really the operational insight. By eliminating the mesenchyme, they eliminated a major source of experimental variability. They removed the need for expensive and complex cell sorting, and they created an ideal platform for studying epithelial intrinsic defects, which covers the vast majority of intestinal diseases that involve mutations in the lining itself.
That purity is foundational. But now they have a pure product. The next challenge is making sure that product isn't generic. It needs to reflect the functional geography of the actual gut.
This moves us into the beautiful complexity of developmental geography. You know, the human gut isn't just a single type of tissue. It has these regional specializations from the top, the proximal end, to the bottom, the distal end.
Right.
The duodenum and jejunum, that's the proximal small intestine, they're responsible for the heavy lifting of nutrient breakdown and absorption. The colon, the distal gut, is mainly about water reabsorption and waste storage. To be a useful model, the HIOs have to mimic one or the other.
And the researchers discovered that these pure MFHIOs could be programmed towards specific regional identities simply by tweaking the specialized cocktail of the 3D culture media. It sounds like they're using the meaty composition as a dial to control the cell's ultimate fate.
Precisely. They tested two specific media conditions to push the identity toward either the small intestine, so proximal, or the colon distal.
Let's start with the one aimed at the proximal small intestine identity.
Okay, so this was the IM plus CK condition. That's intestinal medium supplemented with CHIR to activate WENT and KGF, or keratinocyte growth factor. The addition of KGF was really key here. It generated the most robust organoid outgrowth and successfully promoted that crucial proximal identity. So it was mimicking the duodenum.
And how did they confirm that regional identity? What were the molecular signposts that said, yes, I am a small intestine?
They looked at the transcription factors that act as identity markers. The IM plus CKHIOs showed high expression of general intestinal markers, sure, but critically, they had high expression of PDX1 and GETA4.
PDX1, okay.
PDX1 is absolutely essential for the development of the duodenum and nearby structures like the pancreas. Its presence is a very strong signature of that upper gastrointestinal tract functionality. They also showed low expression of the markers associated with the colon, like SATB2 and MUC2, which is a colonic mucin.
That's a clean result. Successful programming toward the small intestine, specifically the absorptive, enzyme-heavy duodenum. So what happened when they wanted a more distal or colonic identity?
That required the other specialized cocktail, the CK plus DCI condition. This used CHR and KGF along with a mix of compounds, dexamethasone, CMP, and IBMX. This protocol pushed the cells toward the posterior or colonic fate. These organoids express significantly more SATB2 and MUC2, which confirmed a shift toward a more distal, colon-like identity.
But the source material noted that the CK plus DCI condition generated a more heterogene population, correct? It wasn't as laser-focused as the proximal condition.
That's a fair observation, yeah. While it successfully shifted them toward the distal gut, the gene expression data also showed markers for other nearby endoderm-derived tissues like the liver serpin-1, HNF4A, and even a stomach PGC.
Okay.
This implies that the IM plus CK protocol is superior for achieving a specific, homogeneous small intestinal identity. If you need a high-fidelity duodenum, IM plus CK is the recipe. If you need something more generalized or colonic, CK plus DCI gives you that posterior identity, but with slightly more variability.
But despite these regional differences, proximal versus colonic, the key success was that both of these MFHIO protocols still achieved the full cellular diversity we said was critically missing in the old CACO2 standard.
Absolutely. That foundational success was maintained regardless of the regional dialing. They confirmed the presence of all the necessary differentiated epithelial cell types. You had the absorptive enterocytes, which were vilin-expressing, the crucial secretory PANF cells, identified by lysozyme or LYZ positivity.
The source of that antimicrobial defense.
Right, and the hormone-secreting enteroendocrine cells, identified by chromogranin A or CHGA positivity. And in the distal context, they also saw a robust production of mucin 2, MUC2, which confirmed the presence and function of goblet cells.
So this combination of purity, regional control, and full cellular diversity moves us beyond just simple developmental modeling. This is a functional replica ready to be deployed for personalized medicine.
This application is perhaps the most exciting demonstration of iPSCs. Since we can derive these cells from a specific patient, we can create a patient's organ in a dish, complete with their unique genetic defects.
And the researchers chose cystic fibrosis?
Yes, CF, because it's a monogenic disorder, it's caused by a single gene defect. And the affected protein, CFTR, is highly expressed and functionally critical in the intestinal epithelium. It's a perfect test case.
So they're taking patient cells that harbor the specific CF mutation and growing a tiny 3D gut from them. Which cell lines did they compare?
They used iPSC lines derived from CF patients, specifically focusing on the most common and severe mutation, the delta F5038 defect. They compared the delta F508 mutant HIOs against two controls, a naturally occurring wild type or WT HIO line, and crucially a gene-corrected delta F508 IIPIO line.
Ah, that gene-corrected line is the perfect internal control. So it has the same genetic background as the patient, but the single CF gene defect has been repaired. So any functional difference you see is directly attributable to that one mutation.
Precisely. It eliminates all the confounding factors. Now, the challenge. How do you measure the function of the CFTR protein, a tiny chloride channel, in a complex 3D sphere that is only a few hundred micrometers across?
Yeah, that requires an incredibly elegant functional assessment.
It does. They adapted the forskolin-induced swelling assay, or FIS assay, a technique previously validated for use in primary rectal organoids. The principle relies on basic cell physiology and physics. The CFTR protein is a channel that regulates the movement of chloride ions in water across the epithelial surface.
And what role does forskolin play?
Forskolin is the molecular key. It's a compound that activates the CFTR channel. If the protein is working, the channel opens, allowing chloride ions to efflux out of the cells and into the confined space of the organoid's lumen.
And where chloride goes, water follows, due to osmotic pressure.
Exactly. So if the CFTR protein is healthy and functional, the organoid should visibly swell up over 24 hours due to water intake. If it's defective, as it is in CF, the organoid should remain small.
That's the entire brilliance of the assay, using simple water pressure to generate a macro-level quantifiable readout of a molecular-level defect.
And before they even added the forskolin, they noticed baseline physical differences.
Oh, really? What were those inherent differences?
They measured the steady-state lumen area, or SLA, which is the ratio of the internal cavity to the whole organoid. The delta F5081 mutant HIOs had a significantly smaller baseline size and a lower SLA compared to the WT and the gene-corrected HIOs. This suggests the CFA defect is already subtly manifesting in the physical architecture of the organoid even before activation.
Okay, so the stage is set. What happened when they hit them with the forskolin?
A functional readout was just unmistakable. The wild-type HIOs and the gene-corrected delta F5008 or HIOs both showed a significant and robust response. Their cross-sectional area roughly doubled after 24 hours of treatment. It reached about 2.9 normalized units. That swelling confirmed their CFTR channels were functional and allowing the necessary water efflux.
In the patient-specific mutant model, the delta F5081 line.
The delta F5008 HIOs showed virtually no swelling at all. Their mean cross-sectional area remained almost unchanged at 1.073 normalized units. This provides a clear, undeniable functional demonstration of the delta F508 readout in a patient-specific 3D context.
You have to pause and marvel at the elegance of that system. It successfully validates the iPSC-derived mesenchyme-free HIO as a platform for personalized diagnostics. You can imagine taking an individual CF patient, growing their HIOs, and then exposing them to various drug modulators currently on the market.
The ones designed to fix the folding or function of that CFTR protein.
Right. You can literally see which drug makes their personal organoid swell.
That's the future of therapeutic screening for these monogenic disorders. It moves beyond just a binary diagnosis of do you have CF to answering the critical question. Will this specific drug work for your specific mutation and your genetic background? This validates the HIO platform as a powerful tool for disease modeling and drug response prediction, far exceeding the capabilities of any traditional cell line.
So the 3D HIO is fantastic for mechanism of disease and patient-specific validation, but we hinted earlier that 3D structures have a significant limitation when it comes to industrial scale application. If a drug company needs to test, say, 10,000 compounds, manipulating tiny closed 3D spheres is just too cumbersome.
That's the bottleneck. The 3D HIOs mimic in vivo architecture beautifully, but their geometry is just assay hostile. The apical surface, the side where absorption happens and where you need to introduce the drugs, it faces inward, it's locked inside that sphere. It makes it impossible to perform high-throughput dosing or conventional transport assays like those that are standard in a Transwell system.
So we need to flatten the sphere without losing the magic inside. That leads us to the next phase of this revolution, moving from 3D spheres to scalable, functional 2D plates.
The entire pharmaceutical workflow relies on flat, easy-to-access monolayers for high-throughput screening. The massive challenge was creating a 2D sheet that retains the full cell diversity, the structural polarization, and the metabolic complexity of the 3D organoid. Historically, when you try to flatten a complex 3D structure, it just reverts to a simpler, less functional state like CACO2.
So the researchers developed this reproducible two-step protocol to meet this challenge. First, generate highly expandable, immature human intestinal epithelial cells, or HIECs, and then coax them into a terminally differentiated functional HIEC monolayer. Let's break down that path, starting with generating the mass quantity of progenitors.
Right, so step one is progenitor generation. They start with the HPSCs, differentiate them into hindgut cells, and then culture them in what they call HIEC medium 1. This medium contains two crucial components that dictate proliferation and initial fate, R-spondin 1 and insulin.
R-spondin 1 is the key player here, right?
Okay.
It's a really powerful booster of the Renin signaling pathway.
Correct. You can think of WANT signaling as the keep dividing signal for intestinal stem cells, the ISCs. By strengthening WANT signaling using R-spondin 1, they dramatically enhance the intestinal stem cell population, which you can see by markers like LGR5 and ASCL2. This keeps the cells highly proliferative and expandable. They're immature, but they are a massive, sustainable stock.
And the insulin addition in this phase.
Insulin specifically pushes the cells toward the enterocyte fate. It increases the expression of early absorptive cell markers like VL1 and ANPEP. So the outcome of step 1 is a population of immature HIEC progenitors that are highly proliferative, infinitely expandable, and crucially, they can be cryopreserved. You can freeze them and bank them, which allows for true industrial scalability.
So step 1 gets you the scalable stem-like raw material. Step 2 is about hitting the brakes on proliferation and locking in that terminal specialization, getting that full cell diversity we talked about earlier.
That's the terminal differentiation phase. They switch the cells to HIEC medium 2, and the chemical cues completely change. They introduce the Wnt signaling inhibitor 1TC59, and at the same time, they activate the Notch signaling pathway using the agonist valproic acid, or VPA.
So they're flipping two major switches simultaneously. They turn Wnt off with 1TC59 stopping the keep dividing signal, and they turn Notch on with VPA. What is the overall developmental logic of that combination?
Well, in the natural gut, Wnt fuels the proliferation of stem cells, while the Notch pathway drives the commitment of cells toward a secretory lineage, so Paneth cells, goblet cells, enteroendocrine cells. By hitting the brakes on Wnt, they suppress the intestinal stem cell markers. By activating Notch, they push the progenitors to specialize into these mature, functioning epithelial cell types.
So it's a push and a pull.
Exactly. This shift significantly increases the specialization markers like CDX2, vilin, VIL1, lysozyme, LYZ, mucin2, MUC2, and chromogranin A, CHGA. They lose their stemness and become highly functional, mature cells.
This sounds like the perfect system. A highly flexible two-step process. And the research demonstrated they can use this protocol to convert the original 3D HIOs into 2D monolayers, offering maximum flexibility for research.
Exactly. They successfully demonstrated that if you start with the ideal 3D HIO, you can dissociate it, expand it into a spheroid stage they called insects, and then subsequently plate it to form the 2D functional monolayer. This conversion proved that the protocol maintains the maturation status of the HIOs even when the physical architecture is flattened for high-throughput use.
We've established that the 2D functional HIEC monolayer is highly specialized and scalable. Now we need to prove its superiority. We need metrics that show it is functionally closer to the human gut than the old CACO2 standard.
The first functional test is the most fundamental, barrier integrity. How tightly sealed is this epithelial sheet? They measured transepithelial electrical resistance, or TEER, which is the standard measure of how well a monolayer acts as a barrier.
What were the numbers they achieved?
The functional HIECs formed a properly polarized monolayer with significantly higher TEER values than their immature counterparts. The functional HIEs reached 238.56 ohm times centimeter squared. The immature progenitors were nearly half that value. This shows the terminal differentiation step is essential for forming a strong barrier.
Now, how does that compare to natural tissue, and why is the number important?
Well, CACO2 typically forms a ridiculously tight barrier, often exceeding 350. The natural human small intestine, which is designed to be highly permeable for nutrient uptake, typically ranges from 40 to 100. The functional HIECs, at around 238, are clearly much tighter than the natural small intestine, but they are a massive improvement over the immature cells, and crucially, they are still closer to physiological relevance than CACO2, which is often considered too impermeable. They strike a useful balance.
And beyond just the barrier integrity, they also confirmed full structural and cellular polarity, making sure the cell's internal machinery is oriented correctly.
Absolutely. Immunofluorescence confirmed proper polarization. The absorptive brush border proteins like VAL1 were correctly located on the apical or top surface, and the basolateral pumps like Na plus K plus ATPase were oriented correctly toward the blood side of the transwell plate.
But I love the visual evidence of functionality they captured. What did the high-resolution microscopic imaging tell us?
The scanning electron microscopy, or SEM, was compelling. It revealed these irregular blobs appearing on the apical surface of the functional HIECs. These were not seen on the CACO2 cells, and they strongly appeared to be mucus actively extruded from the goblet cells.
Wow.
This is visual, dynamic proof of active, specialized physiological function that the old model simply couldn't replicate.
And the composition of the sheet confirmed the success. of the two-step protocol in maintaining diversity.
Yes. They had the full complement, approximately 69% via L1 plus enterocytes, about 14% MUC2 plus goblet cells, about 2% LYZ plus Paneth cells, and nearly 2% CHGA plus enteroendocrine cells. That full multicellular ecosystem is critical for true physiological mimicry.
Now, let's talk about the metabolic superpower, the specific failing of the CACO2 model. Did the functional HIECs achieve high CYP3A4 activity? This is the real deal breaker.
This is the revolutionary finding of the 2D model. The functional HIECs showed considerably high expression of the drug-metabolizing enzyme CYP3A4. We're talking about levels comparable to what you find in adult human small intestine tissue. The percentage of cells expressing the enzyme was dramatically higher than the negligible levels found in CACO2 cells.
That one difference fundamentally alters the utility of this model for the entire pharmaceutical industry. You can finally model first-pass metabolism accurately in vitro.
And the functional enhancements were extensive, demonstrating true physiological complexity. They measured high activity of intestinal alkaline phosphatase, or IAP, a crucial mucosal defense factor involved in detoxification. They observed increased expression of glucose transporters, SGLT, GLUT2, GLUT5, which is crucial for nutrient uptake, and this translated into enhanced functional sensitivity. They showed significant calcium release in response to glucose stimulation, a hallmark of nutrient sensing.
They even went a step further and showed that this highly functional epithelial sheet could interact properly with external factors like the microbiome and immune cells.
Yes. Due to the high presence of mucins secreted by the goblet cells, the functional HIECs showed significantly higher adhesion of M-cherry expressing Lactobacillus plantarum compared to the CACO2 cells. Furthermore, they demonstrated interactions with immune components. When the barrier was exposed to a bacterial trigger like LPS, a higher number of macrophages attached to and successfully translocated across the basolateral side of the monolayer.
So it's mimicking host defense.
Exactly. This suggests the model accurately represents complex host defense functions that involve communication between the epithelium and underlying immune cells.
Finally, we need to touch on the safety profile, which is crucial if these cells are ever going to be used in advanced tissue engineering or transplantation concepts. They check the in vivo residual capacity of these differentiated cells.
This is a vital regulatory hurdle. Highly differentiated cells are supposed to have restricted, finite proliferative potential. Immature or progenitor cells, by contrast, carry a risk of forming tumors.
All right.
They transplanted both immature and functional HIECs into mice. The immature HIECs retained human cells after 6 to 10 weeks and formed palpable masses containing proliferative markers.
So the immature cells still carried that dangerous proliferative capacity. What about the terminally differentiated functional HIECs?
They were completely different. When the functional HIEC was transplanted, none of the plugs contained any residual human cells after prolonged in vivo cultivation. This is critical validation. Achieving a high state of terminal differentiation, as defined by this two-step protocol, generates a safer product with reduced long-term residual cell capacity. It demonstrates that the one-off notch-on approach successfully locks the cells into a mature, stable fate.
That is a staggering list of functional upgrades. We have scalability, structural integrity, full cell diversity, superior metabolism, enhanced functionality, and a proven safety profile. Now, let's see the ultimate test, using this model to predict how drugs will behave in a real human body.
The immediate translational application of the functional HIEC model is the prediction of the human fraction absorbed, or intestine. As we noted, intestinal first-pass metabolism, specifically that carried out by CYP3A4, is a major factor that the CACO2 model simply missed.
To prove the superiority of the HICs, they needed a clean test case to show that their high CYP3A4 activity actually performs the required chemistry.
They chose nifedipine, a drug that is extensively metabolized by CYP3A4 into a specific metabolite, dihydronifedipine. This allows for a really clear before and after comparison. The functional HAEs demonstrated profoundly robust metabolic activity, increasing the concentration of the metabolite by approximately 7.4-fold compared to the CACO2 cells.
A 7.4-fold increase in metabolic product. That is an overwhelming quantitative failure of the old standard and a huge victory for the new one.
And they confirmed this activity was specific to the enzyme they were targeting by using a specific CYP3A4 inhibitor, ketoconazole. When they pre-treated the HIECs with ketoconazole, the metabolism of nifedipine was significantly inhibited by over 62.5%.
Okay.
This proved the metabolic pathway was both high-level and specific to CYP3A4.
Hmm.
CACO2 cells, conversely, showed almost no change when ketoconazole was added because they had negligible enzyme activity to begin with.
That evidence is compelling. The final, most crucial test was using the HIEC system to predict actual human drug absorption, using drugs categorized by the Biopharmaceutical Classification System, or BCS, which classifies drugs based on their solubility and permeability.
They tested a panel of 6 drugs covering the whole spectrum of BCS categories, from drugs with high solubility and high permeability, so BCSI and 2, to the most difficult drugs with low solubility and low permeability, BCSIV, like furosemide and erythromycin. They calculated the apparent permeability coefficient, or PhD, for these drugs using the HIEC monolayers pleated in the transwell system.
And how did their predictive data correlate with known human data?
The overall correlation was strong. The functional HIEC model showed a good overall correlation with the actual reported human fraction absorbed, the intestinal. This correlation was demonstrably better than the poor correlation achieved by CACO2 cells, especially when looking at those challenging low permeability compounds.
This is where the improved barrier function and high diversity of the HIECs must have provided a critical advantage. What was the practical failure point of CACO2 when testing those difficult BCSIV compounds?
For drugs like furosemide and erythromycin, the CACO2 monolayer was just too tight and too simple. The drug concentration that was transported across the CACO2 monolayer in the assay was often so low that it fell below the limit of quantification, the LLQ.
So they couldn't even measure it.
In simple terms, CACO2 couldn't measure the slow transport accurately enough because it was such a poor physiological mimic. It's too tight, it's lacking the proper transporters and diversity, so it's blind to the absorption characteristics of the most problematic drug class.
Whereas the HIECs were not blind.
Exactly. Functional HIECs, however, successfully determine the drug concentration and the PAL-off for these difficult compounds. This suggests that the HIEC model is exponentially superior for assaying drugs with low permeability, the class where many novel therapeutics often fall, and it addresses a major hurdle in current drug development.
So to summarize the drug screening application, the functional HIEC system is proposed as the superior in vitro small intestinal model because it perfectly combines physiological relevance, full cell diversity, proper polarization, and most importantly, high CYP3A4 activity, which significantly improves the predictive power of drug bioavailability and first-pass metabolism compared to the antiquated CACO2 standard. This means better, faster, and cheaper drug development down the line. What an extraordinary journey into engineering the miniature gut.
It really is.
We've traced the path from the limitations of the antiquated CACO2 standard to the dual breakthrough enabled by iPSCs.
We saw the development of pure, mesenchyme-free 3D HIOs, the small, complex ecosystem perfectly suited for dissecting detailed disease mechanisms, demonstrated so powerfully through that Delta F508 cystic fibrosis model and its elegant swelling assay. This is the model you use when you need to understand the exact mechanics of an epithelial intrinsic defect in a patient-specific context.
And complementing that 3D sphere is the highly functional 2D HIEC monolayer. This is the scalable, high-throughput solution that maintains critical cellular diversity and, crucially, achieves high activity of the drug-metabolizing enzyme CYP3A4, allowing for accurate prediction of oral drug bioavailability, a massive win for the translational side of drug development.
This represents a tectonic shift in scientific capability. We are moving from generalized, biologically simple cell lines that offered limited predictive value to highly defined, patient-specific models. Instead of just hoping a drug works based on generic data, researchers can now test therapeutic compounds directly against an individual's personal gut tissue architecture. This is truly paving the way for personalized medicine, particularly for complex gastrointestinal disorders like IBD or specific absorption disorders.
And the research itself even points to the next challenge. While these functional HIECs are superior, they still slightly differ from the in vivo human intestine. Specifically, they noted that the barrier function, measured by TEER, is a bit higher than natural tissue.
Right. They're pure epithelial sheets, which, while fantastic for studying intrinsic defects, lack the complex underlying support structure, the mesenchyme, the vasculature, and the immune components, which naturally regulate the final maturation and permeability in vivo.
So given the progress we've seen in successfully generating these defined 2D monolayers, what happens when we intentionally start reintegrating those missing components, not as contamination, but as controlled necessary inputs into these scalable 2D systems?
That is the next great frontier. The pursuit of in vitro perfection will almost certainly lead to even more accurate organ-on-a-chip platforms, where controlled microfluidics and structured co-culture of epithelial, mesenchymal, and perhaps even vascular cells further close that functional gap.
But there's a trade-off.
The challenge will be maintaining the assay-friendly simplicity that these 2D platforms have achieved while layering on the necessary complexity to reach true in vivo fidelity.
It's a delicate balance that dictates the immediate future of precision medicine in gastroenterology. How simple can a model be while remaining fully predictive? That's the question for the engineers of the microgut revolution.
Indeed. It seems the small intestine still has a few secrets left to give up.
A challenge for the next deep dive. Thank you for joining us on the Deep Dive.